Good time to everyone. I will discuss the alterations in neuroplasticity caused by depression and chronic pain and how transcranial direct current stimulation, tDCS, offers a promising neuromodulatory approach for treatment. Additionally, I will highlight the crucial role of mitochondria in these processes and explain how depression and chronic pain share overlapping pathways of neuroplasticity disruption. Neuroplasticity, or the brain's ability to reorganize itself, is essential for learning, memory, and adaptation. However, in depression and chronic pain, neuroplasticity becomes dysregulated, leading to persistent symptoms and functional impairments. Let's have a short look in neuroplasticity alterations in depression and chronic pain. Both depression and chronic pain involve maladaptive synaptic plasticity, neurotransmitter imbalances, altered brain connectivity, neuroinflammation, and mitochondrial dysfunction. Depression is associated with reduced synaptic strength, particularly in regions like the prefrontal cortex, hippocampus, and anterior cingulate cortex, leading to impaired emotional regulation and cognitive deficits. It also involves low levels of brain-derived neurotrophic factor, BDNF, which is critical for synaptic growth and resilience. Additionally, glial cell dysfunction and neuroinflammation contribute to neural damage and impaired plasticity. Chronic pain leads to central sensitization, where neurons in pain processing regions become hyperexcitable. Chronic pain enhances long-term potentiation in pain pathways, strengthens pain-related synapses, and thus making pain persistent. It also induces neurotransmitter imbalance, particularly increased glutamate and decreased GABA, heightens neuronal excitability, and thus prolonging pain perception. In chronic pain, similar to depression, glial cell activation and neuroinflammation exacerbate neuronal dysfunction. A key similarity is that both conditions show decreased gray matter volume in pain and mood-regulating areas, contributing to sustained symptoms. And let's have a short look in mitochondrial dysfunction. As a common factor, mitochondria play a fundamental role in neuroplasticity by supplying ATP for synaptic function and regulating oxidative stress. In depression, mitochondrial dysfunction leads to energy deficits, increased oxidative stress, and reduced neurogenesis, exacerbating symptoms. In chronic pain, mitochondrial impairments in neurons contribute to persistent hyperexcitability and impaired synaptic modulation, making pain more resistant to regulation. Depression and chronic pain involve mitochondrial DNA damage, impaired calcium homeostasis, and increased inflammatory signaling, and thus further worsening neuroplasticity deficits. What is the action of tDCS? Transcranial direct current stimulation acts as a neuromodulatory treatment. tDCS is a non-invasive brain stimulation technique that modulates cortical excitability and promotes neuroplasticity through weak direct electrical currents. Its therapeutic effects in depression and chronic pain are achieved via multiple mechanisms. The first effect is synaptic modulation. tDCS enhances long-term potentiation in hyperactive regions, for example, prefrontal cortex, in depression, and restores inhibitory control over pain pathways in chronic pain. The second effect is neurotransmitter balance. It increases glutamatergic and dopaminergic transmission in mood-related circuits while restoring GABAergic inhibition in pain circuits. Another effect of tDCS is upregulation of brain-derived neurotrophic factor, tDCS stimulates BDNF expression, promoting synaptic repair and neurogenesis. And the other effect is neuroinflammation reduction. It decreases glial activation and pro-inflammatory cytokines, reducing neuronal damage. And the most important is the potential mitochondrial enhancement. tDCS has been shown to improve mitochondrial ATP production and reduce oxidative stress, and also enhance cellular energy efficiency, which supports neuroplasticity recovery. Now, I will share the effects of tDCS in modulating chronic pain and depression in a number of our patients. A randomized prospective study was conducted on 214 patients suffering from primary chronic pain and fibromyalgia that both were grouped together under chronic pain. The patients were divided into 4 groups. Group A received active tDCS. Group B received active tDCS combined with SSRIs, mainly 50 mg of sertraline. And Group C received only SSRIs, and the control group of sham tDCS, and the pain duration of 36 weeks. Patients were assessed for pain intensity using a numeric 0 to 10 pen rating scale and depression severity, Beck Depression Inventory rating scale from 0 to 63. The groups A and B underwent 6 tDCS sessions over 3 consecutive weeks. The anodal stimulation with 2 mA intensity was targeted at the primary motor cortex and applied for 20 minutes per session. The control group was included, consisting of 10 randomly selected patients who received only 2 sessions of sham tDCS. Our assessments were conducted at 3 time points: at baseline, after 2 sessions of tDCS, and after 1 month of treatment. The primary outcome measures included pain and mood changes, with depression levels assessed using the Beck Depression Inventory. Our results: a significant improvement in both mood and pain scores was observed in both groups A and B with active tDCS compared to sham and SSRI groups. Group B with adjunct therapy showed most significant improvement in Beck depression scores, along with better daily functional changes compared to the other groups. The Group C, SSRI group, showed some improvement, but it was not as significant as the groups using tDCS. This study suggests that tDCS, particularly when combined with SSRIs, provides significant benefits in reducing pain and depressive symptoms in patients with chronic pain conditions. Here, I would like to mention that Professor Stavros Balogiannis always has emphasized on the role of mitochondrion. He has published enough books and articles sharing his own experiences studying mitochondrion by microscope. Professor Balogiannis explained mitochondrion as a unique organelle that contains its own genome, encoding essential proteins that are major components of the respiratory chain and energy production system. Mitochondria play a dominant role in the life and function of neurons and glia. Grateful for the legacy Professor Balogiannis left behind. Various studies utilizing positron emission tomography and single photon emission tomography have demonstrated reduced blood flow and metabolic activity in brain, particularly within the frontal region in patients suffering from both chronic pain and depression. These findings suggest that there may be overlapping functional anatomy in the nociceptive and affective pathways also. Various studies, including Sang-Bin Lee and colleagues, have reported anodal tDCS tended to stabilize the mitochondrial condition in mice. By addressing these shared pathways, tDCS emerges as a potentially effective intervention for both conditions, paving the way for better non-invasive treatment strategies in neuropsychiatric and pain disorders. And at the end, I would like to extend my heartfelt thanks to the Professor Chalaki and the organizing committee for such a fruitful congress that has brought us all together, fostering insightful discussions and collaborations. And thank you very much for your time.